Hi, this is lecture 4 for Biosci 101, spring 2021 by Dr. Giorgi, and today's topic is GLP. What is that acronym? That means good lab practices, just basically customs in lab that you should follow because they're important. So maybe more than custom practices, as the name says. It's hard to rename something when it's sort of this clear of a name. So good lab practices. By the way, this is an important thing to put on your resume. It's something especially that industry likes to see. It's one of those buzzwords that the computer will screen your resume for. So do you know GLP? Yes, you know GLP. In fact, here's some official training today. All right, we're going to start with the most important GLP of all, which is lab safety. Safety is always the first thing that you should think of anytime you're doing anything in lab. And my main rule for safety is just be aware. Be aware of where your body is, which in lab, by the way, means you always move slowly. What looks like a fast walk to the rest of the world is actually a run in a lab, which only happens if it's an emergency. So move slowly and know where your feet are, know where your elbows are, know where your hair is if it's long. Just know where each part of your body is, be aware of it at all times, and then know what's around you and who is around you, who is near you. Don't bump into people or things. And really key is to know what you're using. Is it electrical equipment? How can it harm you? How can you harm it? Is it a chemical? There's something called an MSDS, Material Safety Data Sheet. You can look up if you don't know what it is. They'll tell you the worst case scenario. If you look up sodium chloride, salt, table salt, it'll tell you the ways it can kill a person. But like most poisons, everything's in the dose, okay? So learn about what you're using, what you're touching, what equipment you're using, what supplies you're using, and ask questions. There's nothing more important than safety. And the number one rule around safety is make sure the humans are safe. The second thing is make sure the expensive equipment or the really precious experiments are safe. But number one is be safe yourself. Here's some fun graphics to remind you. We don't eat in lab. Um, I don't know that we can dispute whether eating all these hot dogs is a good idea or not, but definitely don't do them in lab. The idea is you're always assuming that something's toxic because you don't know, you didn't watch that surface for 24 hours a day. And so, although these days we kind of do, but usually lab surfaces and lab items are shared, and assume that somebody spilled something toxic and they didn't clean it up. Prepare for the worst. And basically, you never eat in lab. You barely drink water if you really must, but you go out into the hallway to eat or drink, or even just don't put anything in your mouth. I think we're maybe more aware of this whole thing that our mouths, our faces are vulnerable now. Our skin protects us beautifully, except where there's holes, like where things are supposed to come in, like our nose and our eyes, of course, and our mouth. So just be careful around those parts. And anyway, you're wearing two masks right now if you're in lab. And so no shenanigans like this. It's fine in a hallway, but not in a hallway in your lab. Assume that things are dangerous until you know they're not in lab. And the good news in microscopy is very few things are dangerous. But nonetheless, be careful. There's always something that can go wrong. And so this is actually some slides I stole from another instructor who, you know, ideally this is not your lab bench, but in truth, this is the lab bench of a lot of really successful scientists. As long as it's organized and clear to you, that's really important. But the less clutter, the more organization, the better. This bench is mostly, it's not so much disorganized as there's not a lot of room, but it might just be they don't have a lot of room. And notice the lab notebook, that's a typical lab notebook right there. So, you know, just again, be aware, know what's going on. This is an old school version of PPE, but of course, these days we're talking, well, face shields and double masks. And because things can drop on our feet and we might not notice them, we don't wear open toe shoes like flip-flops to lab. Sneakers are fine. And you know, so we try to cover up just in case so things don't land on us and we don't have to depend on our skin. Um, there's information about what you're dealing with, and it should be posted in the lab. And um, this will, like, these kind of symbols tell you all sorts of, your danger levels. Basically, we don't have any of this. Our scopes are not, hopefully not flammable for the most part. And they're not biohazards either, obviously. However, our specimens are biohazard. So this symbol is where you collect waste, and you're going to do your cheek cells at home. If we were doing it at Merit, we would collect your cheek cells in a biohazard waste bag. You can, maybe now that we're thinking about infectious diseases, we realize why. So obviously, if you're living with yourself, which you are, you're not a biohazard to yourself mostly, but humans are considered, human specimens of any sort are considered biohazards to other humans, as indeed we have now seen thanks to the pandemic. Okay, so switching gears, here's some general info about good lab practices in microscopy in particular. So there are three parts to the microscopy process. There is the specimen prep, so making the specimen. This can be an entire career, like being a histotech. That's what you're doing. You're making the specimen in a way that is really difficult and really powerful to do. Then once the specimen is made, and sometimes specimen prep is as easy as drop a fly, there's a fruit fly, catch it, put it on a slide, put some liquid, a cover slip, and there, 2 second specimen prep. Other times it can take weeks if you're preparing it for EM. So it just depends. But then once you've made your specimen, you're ready to acquire it. So this is where you're sitting at the scope and acquiring, getting an image of your specimen. And this is where you need to understand the scope and the parts of the scope and the software, because pretty much we're going to assume the scope is digital. There's a camera and there's software attached that you're going to have to figure out. So there's much to learn about image acquisition. You're going to learn about all three of these in this program. Once you've acquired the image and it's on a flash drive, because most scopes are not network because the administrators don't want to deal with viruses actually. It's on your flash drive, and then you've taken it to 2 or 3 other locations, your computer, the cloud, etc. Now you're talking about image processing and, of course, analyzing the data that you got from the scope. And those three parts really take about, each of them takes, generally speaking, as much time, and certainly there's as much knowledge to be had about specimen prep as there is about image acquisition, as there is about post-image acquisition, post-acquisition image processing. And again, these are things that you tell future employers, I can do this. I understand image processing. And they're like, wow, it's a thing. And that there's a whole world of what's correct and what can be done and what cannot be done and so on. And you know how to analyze data, that images are data and that there's lots of info to be gotten out of them for science. Okay, so the fundamental is do everything the best you can. Each of those 3 steps should be as good as you can possibly do it. There is a tendency that a lot of people have to just skimp on the specimen prep and go to the part where you're sitting on the scope and figure out the scope's really expensive. It'll fix magically anything I do. Actually, I don't think any of you will be succumbing to this, especially those of you who are histotechs. You know the value of a good specimen prep. But if you end up working with a bunch of scientists who aren't trained on microscopy, you'll find that there's a huge tendency to figure out, to sort of count on the $500,000 confocal to fix anything, any shortcuts they took in specimen prep. And it just doesn't work that way. So garbage in, garbage out. So don't, don't think I can skip step one, specimen prep, and step two, image acquisition. I don't have to do them that well because I'll just fix it in Photoshop later. No, no, that's not science and that's not good data. So do each step as well as you possibly can. And and again, just reiterating this, prepare your specimen well, and then also acquire it beautifully. Sit there and look at the dynamic range. You just learned about that. Make sure it's not saturated. Make sure you've got the histogram up and you can see what's going on with your specimen. Put the little, check the saturation warning, lookup table. It's super useful. Make sure it's focused, make sure there's good contrast, etc. You know, look at what you're imaging, and don't be shy about acquiring a lot of images. If you're kind of going, well, maybe it'd be better this way. Oh, let me try it this way. Do it all. We got space on a computer. It's just numbers. It's just a grid of numbers. So your computer can handle it. Once we start talking about movies, you start running into space issues, but just digital images, go for it. Your the image that you actually snap, that is in the software of the camera, or the driver of the camera, basically. That's called your raw image, and it's data. And that's considered, this is the thing that happened. This is the ground truth, if you will. This is the starting point. And of course, we know now that you can sit at the computer and manipulate the software because it's a digital image, and you can make your original raw data look 10 different ways on the same scope just by manipulating parameters. Nonetheless, those images that come off of the scope itself are considered your raw data, your raw image, and then anything you do after that is considered processing it. So then if you take it to more software off of the scope and then do things you could have done at the scope, well, now it's considered processing the image. And it's all legit, and it's all stuff that you need to keep track of. So this is a hugely important, fundamental, good lab practice in science. Whatever you're doing, document it, write it down, document it with, this point in time, with notes, not so much videos. We don't record everything we've done. We have a notebook and we write down every single thing we've done. If you're not sure if it needs to go in the notebook, put it in the notebook, because it's always better to have too fat of a notebook than to come back 3 years later and go, oh, what did I do that day? That was the day the cells did the amazing thing that I now know could cure cancer, but what did I do? Why didn't I write it down? Okay, that's the mindset of a scientist. Ever since I started doing science for real, like when I was a lab tech, I have this habit of recording. It's funny, it like spills over into my regular life, but I'm always walking around with a notebook. I'm always doing something and then immediately writing it down. Oh, look, I did that. If in doubt, write it down. But, try to keep clear, organized notes. It's actually a good thing to put your thought process in there. It helps you remember things. It helps you understand why you were doing something. It's okay to have a little bit of context, like on the side, you can say, this is the day this wasn't working, or I was, when I'm taking notes for myself, I even put in things like I'm hungry or what I'm going to eat tonight or whatever. When I'm taking them for a lab notebook, though, it's a legal document, so I don't put my personal life in there, but I do put in things like, there's a weird odor in the lab, or anything like that, anything that might be possibly relevant goes into the lab notebook. And again, more is better. Most people underwrite. Very few people write too much in their lab notebook because most of us just want to get on with it. We want to do it. We don't want to stop and write. And that's another big thing is do not write at the end of the day, you write as you do it. And when you're sitting at a scope, that means you take an image, you record it in your image log. We're going to talk about this in a second. But it doesn't mean you take 50 images and then you go, I took 50 images. Okay, so what's this image log? The image log is literally a list of every image you ever take, every digital image that you ever take. And when you have the scopes at home and you might not have the chance to do digital images, then drawings suffice or records of what you saw, notes basically. But um, the metadata is, so in your image log, you should have a list of every digital image you ever take, including the metadata, which is the technical info of the magnification, the exposure, the fluorophore, things like that. Um, some software will record this for you and even make it viewable to you, but there's a lot of software that when you snap an image, it doesn't save any info about what you just did. And so I keep asking my microscopy folks, like, is there, are you still all writing image logs in a notebook? And they're like, yeah, except for like maybe on a confocal, which you can trust. So it's a great habit to get in. It just allows you, it's literally a list of all the images you took. And that also includes when you take your raw data and then you create a new processed image, you want to put it in there. Okay. So you want to be able to just quickly scan and go like 3 months ago, I have 5 images of this. Okay, let me look them up. Here's their name. There's going to be a handout in your module that gives you an example of exactly what you should include, but it's basically this info. It's in the beginning, you can just have a session info. So, I'm looking at liver on this such and such a date and such a scope, but every image gets its own line, and there's a name, the exposure, the magnification, is it red, green, blue, if fluorescence or phase or bright field or whatever. If you're manipulating gain and offset because you're on campus, and I guess it's not so applicable this semester, but next semester you can do that, you include that too. It's really good to have little quick drawings that are just super, like a little triangle or something, or 2 cells next to each other, etc. I personally find those really useful when I'm scanning thousands of images. I'm like, where's the one with the long cell? Oh, there it is, that kind of thing. And any notes like 3 stars, this was a really great image, or this one I'm just showing for this, any notes to yourself, kind of. Remember, you're basically not going to run. Like, remember, you're not going to remember this, every image you took a few months from now. It may seem that inconceivable. You're like, but I just took the best image of my life. I'll never forget it. But in truth, you will, because you're a human being and most of us will forget it. And we need these notes. And actually, these notes are, when you're in a lab, they stay with the lab and they are legal documents, and patents and billions of dollars have been won over notebooks, by the way. So they're important documents, and someone else should be able to look at it and understand it. That's a key thing. And if someone else can understand your notebook, then so can you 3 months from now, because you're basically a new person. And yes, include this when you take an image out and you change it up and do fun stuff with it on software and do some filters and things like that. Also, by the way, since they are legal documents, this is why lab notebooks, we don't use whiteout. And the idea is you're trying to hide something. If you make a mistake, just draw a line through it. Just be like, ignore this, draw a line. In industry, they're going to ask you to sign and date every page and not to have any blank space. And so if you do have blank space that you left, you're going to draw a line through it and sign your name on that line. Again, you don't want to try and recreate what you did 2 days ago. You do it as you go. Every few minutes, you should be writing in your lab notebook. And if your memory is tremendous, then it can be every 10 minutes or so, but you're doing it simultaneously, basically, and certainly not at the end of the day. Also, yes, it's totally legit still to grab. This happens so much. It's hilarious to grab a paper towel and write your notes on that because you forgot to bring your lab notebook over to the machine, and then you've got 5 minutes to put this in, and so there's a lot of lab notebooks where you can add in things. You can staple them in, you can glue them in, tape them in, anything like that. So real lab notebooks do have basically paper towels with info in them. So processed images, we already talked about that. Always save the raw images, always make a copy before you do anything to your original image. And this is just an example of what you might write when you're imaging. Sometimes it feels like there's not a lot to write, except I sat down, I used the scope, you know, and like you're going to make a line for every image that you captured for your image log. But what about the lab notebook part? Which, by the way, ideally it can be in the same physical book, but you flip it over and one side is the image log, the other side's your lab notebook. So what do you write about an imaging session? Well, anything you can think of to write, you know, of course, what you're doing, but your own thinking is good to write down, even if like, I wasn't sure what I was looking for. And then I came across this really great slide. And then I realized that I was looking at the wrong thing. And, you know, your thought process actually helps you to come back to that time when if you're, you know, 5 years from now, you're like, why did what's this an image of anyway? So, you know, here's some examples. Okay, hopefully there's no earthquakes, but you never know. So at the end of this program, hopefully you have thousands of images. And how do you search back and find this? And again, you're going to probably go to your lab notebook first and then your image log. Just depends on what you're looking for. But this brings us to file names. How are you saving each of those thousands of images? What is the GLP, the good lab practice for image file names in microscopy? Well, there's no consensus. It should be unique, simple, clear, and relatively short because you're writing it in your imaging log a million times. And yes, you can use ditto marks. And pretty much the main thing is to allow for a series tag. So the first image, the second, the third, the fifth, and usually we do a 001, 002, so you can have up to 999 images in that series, you know, 1,000 images of a liver slide or something like that. And eventually you want to pick a system and go with it. It's fine if you're not consistent at first and just trying out what works for you. I say don't put tons of info in the file name because it takes too long to type it in and also record it in your image log. But almost everyone uses their initials as a starting point to ID it as your own. And I kind of recommend going by date because then you can look back in your lab notebook and see what was the project I was doing on that day. That is what most people do. So, and they'll do usually, actually, I would do GG, the date, and then 001, but you can do A to Z, or you can put the date first if you prefer. Anything that makes sense to you and is short and is easy to repeat. Some people like to organize their life and their notebooks and their images by projects. So I tend to do overlapping projects, so I haven't used that, but so there's a project fail typically in most semesters, and so that would be, you know, my project fail images. You can certainly do it by specimen. That's a very common way to label things. So it kind of depends on what you're doing and what makes sense, and just think about it. But you want to be consistent, generally speaking, but honestly, in this class, play around a little and see what works for you. And then if you go get a job in a lab and they're like, this is the way we write the image names, go with it until you're the boss, and then you can change it. So a lot of people like date, as I've said, because your lab notebook is by date. So it just kind of makes it easy to find. file formats of your images. Oh, good Lord. This is a big issue because there are a zillion different file formats because the computer programmers have gone mad and it's a job creation thing for programmers, basically, is the way I see it. So some of the file formats are great and others are pointless. But since the software is also anarchy in microscopy, there's many different types of software. There's many different types of file formats too. And it's a big issue because not all software reads all file formats. Use TIFF. Okay, TIFF is what's called the lossless file format. It has all the info it can possibly have. It's going to be a fatter, bigger format. So your raw data, your original image should be a TIFF. You can then save a JPEG, write it in your image log, make a copy, call it a JPEG. Nobody uses BMPs that much anymore, but JPEGs, now the computers are quicker and bigger and so on. JPEGs work really well. You can use that as a display image, but not something that you're going to process or, alter or anything like that. It's sort of your final image that you put into the PowerPoint that you're going to show to people. So try to use a TIFF when you can, if that's an option. This is a good time to tell you the story of the software in microscopy. So once upon a time in the good old days, there was one software. It was called Metamorph. Metamorph was the name of it. It actually started as Image One, which I used, and then it morphed, it changed into a software called Metamorph, and everybody used it. It was fantastic. It was written by scientists for scientists. It was just so good that a company bought it and put it out of business for no good reason. And because that's just corporate America, things happen. And at that point, all the scope manufacturers decided they would each develop their own software. So the big four manufacturers are Leica, Zeiss, Olympus, and Nikon. Those are the big four manufacturers, especially of these sort of high-end scopes for, high-end users, really. Okay, so those four companies said, we're going to make our own software. And guess what? We're going to make a file format. So when you acquire the image on our scope for using our software, only our software can view that, that format. And so you're going to buy more copies of our software to have on your other computers where, when you're not sitting at the scope, the next person's on the scope, you're going to take your software somewhere else to process it. And then, oh, good, we're going to sell you more of our software. And they did that, and it still happens. And you're going to still run into this. And then there's image processing software. They have their own file formats. It's just ridiculous. There's a lot of file formats. So you might not be able to save something as a TIFF. It depends on the computer and the software and the company and what you're using. But if you can, save it as a TIFF. And if not, then you might have to convert it. Thank goodness there's a group of microscopists who are trying to organize against this, openmicroscopy.org, and they have little software that will convert any image into another image, any format into another format, basically. And there's literally hundreds of imaging formats, as you can see if you look at their site. So it's chaos and anarchy and a waste of time right now, but that's where we're at. That was a while ago, but I bet they're always hiring, they're always getting bigger. And people are working on trying to have universal formats and things like that, but we're just not there yet. TIFF, however, is almost a universal format because most imaging software works with TIFF files. So we love a TIFF image. Okay, so a summary. While acquiring your images, record each one in your image log along with the metadata. Also record the session in your lab notebook. And if you can, if given the option, acquire and save your image as a TIFF. And then this is when you're at school, but this is true on any scope. Like, don't leave the image on the scope. Well, leave it on there and let whoever's running the place, which might be you, clean it off. But assume that somebody's going to come and empty out that computer, which in truth, at Merit, we don't do very often, but you don't want to count on that. You always want to walk away from the scope with your own copy on your own flash drive and then put it 2 or 3 other places in case one place crashes, like on a computer, on the cloud, don't consider your flash drive a place, on another computer, or at least 2 places if you can, 3 is better. Um, this is in the old days, we would literally mail each other flash drives, like me to other scientists in other parts of the country just to save images in case, there's an earthquake or something in California. Now we've got the cloud, so things are better. Always remember to keep a raw version and then copy it before you start playing around and processing it. And record any new images that you've processed and done fun things to in your image log also. That is it for today.